In Chapter 16, we are going to discuss gene expression. For a cell to function properly, necessary proteins must be synthesized at the correct time and at the correct place. All cells control or regulate the synthesis of proteins from information that's encoded in their DNA. The process of turning on a gene to produce RNA and protein is known as gene expression. And whether we're talking about a simple unicellular organism or a complex multicellular organism like a human being, each cell controls when and how its genes are expressed. And for this phenomenon to occur, there must be internal chemical mechanisms that control when a gene is expressed in order to make RNA and protein, and how much of that protein is actually made. And the genes also have to regulate when it's time to stop making that protein if it is no longer needed. So in Chapter 15, we talked about the central dogma. Remember, the central dogma states that DNA is transcribed into mRNA, which is then translated into protein. And we talked about the different steps of transcription and translation and the similarities and differences between them. But the regulation of gene expression, it's really important because it conserves energy and space. It would require a significant amount of energy for an organism to express every single gene all the time. So it's actually more energy efficient to turn the genes on only when they are required. In addition, only expressing a subset of genes in each cell saves space because DNA must be unwound from its tightly coiled structure to transcribe and translate the DNA. Cells would have to be enormous if every protein were expressed in every cell all the time. So the regulation maintains efficiency by regulating energy, regulating space, and regulating time. And this whole intricate control of gene expression is an extremely complex process. And the malfunctions in this process can be detrimental to the cell and can lead to the development of many diseases, including cancer. And in cancer, we have oncogenes and proto-oncogenes. So oncogenes are the bad genes. Those are the cancer-expressing genes. And proto-oncogenes are the good genes. Those are the genes that protect us against cancer. And cancer as a whole, it's a group of anti-apoptotic cells. So cells can either die via two mechanisms. Cells can die via apoptosis or necrosis. And if the cells don't undergo the apoptotic proper programming, they become anti-apoptotic, and they don't die when they should, and they proliferate, and they spread, and that is cancer. And the genetic content of each somatic cell, each cell that is undergoing mitosis in an organism is the same, but not all genes are expressed in every single cell. That would be counterproductive. The control of which genes are expressed actually dictates whether a cell is an eye cell or a liver cell or a pancreatic cell. And it is the differential gene expression patterns that arise in different cells that give rise to the complete organism. The regulation of gene expression occurs at multiple steps, going from DNA to the functional gene product, which is usually a protein. And what we're going to start with is this external signal that is speaking to the chromatin that is located in the nucleus. So the chromatin structure makes the DNA more or less accessible for transcription by RNA polymerase. And in eukaryotic organisms like human beings, the primary mRNA transcript must be processed before it can be translated in the cytoplasm. So transcription and translation actually occur simultaneously in the cytoplasm. Really important bullet point up here. But regulation occurs at the transcriptional level, and translation is exclusive to the cytoplasm. So when we translate, we're translating mRNA into protein. So the final level of the active protein that is expressed in the cell depends not only on the rate of synthesis, but also the rate of the degradation of the mRNA and the protein itself. So I actually studied this mechanism in-depthly during my graduate work at Florida State, where I studied the circadian biological clock. And what we've come to learn about the circadian biological clock is that in circadian biology, the same signals that start the gene expression actually turn off the gene expression. And this is termed a negative feedback loop. So that negative sign, so we write negative feedback loop. So in the circadian biological clock, there's a signal coming from the particular circadian genes, the period and the cryptochrome genes. those genes are going to start the production of mRNA. So mRNA will begin in the nucleus, and then transcription will occur. There's a shuttle, and the mechanism of where transcription actually occurs, there's actually a little bit in the nucleus, but the majority occurs in the cytoplasm. After transcription, we have what's called post-transcriptional modifications, where there are different players that come in and modify the protein, or sorry, the mRNA, such as casein kinases and other regulatory elements. And then after we get the mRNA in the cytoplasm, the mRNA needs to eventually be translated into protein. So the period gene gets transcribed into a transcript, and then that transcript gets translated into the period protein. And that protein becomes active, and a lot of things happen to that protein. That protein undergoes what's known as post-translational modifications. And these post-translational modifications include sumylation, methylation, ubiquitination, deubiquitination, and the protein builds up in our body over the course of the day. But eventually what has to happen is the protein must be degraded. And the protein is actually degraded because there are signals that get sent back to the nucleus over here that actually inhibit the start of the new cycle, actually inhibit the production, the termination phase of the protein synthesis. So once protein synthesis is inhibited, that actually stops the production, and it allows for the cell to reset, and then we can start back here. We can start back with a new signal. So it's a negative feedback loop. The same thing that turns on the reaction turns it off. Negative feedback loop. Really important to understand that transcription and translation are occurring simultaneously in the cytoplasm, and that translation is exclusive to the cytoplasm. This slide is really important to understand some key regulatory differences in gene expression when we talk about what is occurring in prokaryotic organisms versus what is actually occurring in eukaryotic organisms. So again, the big thing is that the prokaryotic organisms lack a true nucleus. The majority of their DNA is found in the nucleoid. And then the eukaryotic organisms, all gene transcription is going to start in the nucleus, where we have a high density of chromatin. So again, the DNA in the prokaryotic organism is going to be found in the cytoplasm, the nucleoid specifically, and in the eukaryotic organisms, the DNA is going to be confined to the nuclear compartment. Prokaryotic organisms, the RNA transcription and protein formation occur almost simultaneously. Eukaryotic organisms are very different because we have to make the mRNA transcript, and we have to have modifications to that mRNA transcript in order for that transcript to be translated into protein. So the translation of RNA to the protein actually occurs in the cytoplasm. Really, really important to know. And in prokaryotic organisms, the gene expression is going to be regulated primarily at the transcriptional level, where the eukaryotic organism gene expression starts from the initial synthesis. So it's regulated at the signal level, it's regulated at the epigenetic level, it's regulated at the initial gene formation, when that gene undergoes modifications, when that gene is being transcribed into mRNA, and the post-transcriptional modifications that happen to it afterwards. And that transcript that is then modified undergoes translation to become a protein, and then there are post-translational modifications to that protein, like I mentioned on the previous slide: sumylation, methylation, ubiquitination, deubiquitination. So this is a really important slide. There's going to be several quiz and test questions coming from this slide. Really important to be able to understand how gene expression regulation differs between prokaryotic and eukaryotic organisms. So we mentioned this on the previous slide, but we're just reinforcing this extremely important concept of where the DNA is located in the prokaryotic organism. So the DNA is located in the cytoplasm, specifically in the nucleoid. And proteins with similar function, or proteins that are found within the same biochemical pathway, they're organized in blocks known as operons. So for example, all the genes that are needed to use lactose as an energy source are coded next to each other in the lactose operon, or the lac operon. And that makes it very simple for the gene to do its proper function, so that the gene can transcribe everything all at once into a single mRNA transcript. And in prokaryotic cells, there are 3 types of regulatory molecules that can affect the expression of operons. So they're listed down here below for you. So first, we have the repressors, which are going to repress or suppress transcription. Then we have the activators. When we have activation, we have an increase in something. So the activators increase transcription. And then we have the inducers. So if we induce something, if we induce inhibitory signals, we can suppress. If we induce activation signals, we can activate. So inducers can do similar function to both the repressors and the activators based upon the necessity of the particular cell at the time. So whatever the cell needs, the inducers are going to be more of the modifying operon. Bacteria such as E. coli need amino acids for their survival, and they're actually able to synthesize many amino acids. Tryptophan is one that E. coli can either ingest from the environment, or they can synthesize using enzymes that are encoded by 5 particular genes. And these genes are next to each other. You can see the genes that are listed here. So these are the 5 genes that E. coli can use: TRPE, TRPD, TRPC, TRPB, and TRPA. So these 5 genes are known as the tryptophan operon. And these genes are transcribed into a single mRNA, which is then translated to produce all 5 enzymes. If tryptophan is present in the environment, then E. coli doesn't need to synthesize it, and the TRP operon is then switched off. However, when tryptophan availability is low, the switch controlling the operon is activated or turned on, and that causes the mRNA to be transcribed. And in turn, the enzyme proteins are going to be translated, and tryptophan will be the resulting protein. So again, this picture is the tryptophan operon. And the 5 genes that are needed to synthesize tryptophan, I already highlighted. So the TRPE, D, C, B, and A. Those are the 5 genes needed to synthesize tryptophan in E. coli. And they are located next to each other. Those are considered the TRP operon together. When tryptophan is plentiful, 2 tryptophan molecules can bind to the repressor protein. So here's tryptophan binding to the repressor right here. And they bind actually at the operator sequence. And what this does is this physically blocks the RNA polymerase. So that's why you see the X here. This is physically blocking the RNA polymerase from transcribing the tryptophan genes. But when tryptophan is absent, the repressor protein does not bind to the operator, and then the genes can be transcribed. And I mentioned this concept on the previous slide, but I just want to go over it again. This is showing you what happens when tryptophan is absent. So when tryptophan is absent, the repressor protein, it doesn't have the proper signals it needs. So the repressor protein does not bind to the operator, and this allows for gene transcription to occur. Again, the previous slide showed when the repressor was bound, and we inhibited that transcription. And here it's showing when the repressor is unbound, and this allows for the genes to be transcribed. So again, 2 important concepts. Genes are transcribed when the repressors are not bound. The repressors are repressing or suppressing that action. We allow for gene transcription. So you can see the operon here, and we are transcribing everything. So in the absence of tryptophan, the repressor, again, is dissociating from the operator, and the RNA synthesis proceeds. So we're able to make the transcript that we need. When the repressor binds, it suppresses this, and we cannot make the RNA transcript that we need. And these processes are very important. So let's say the body is processing or making too much mRNA. Well, then the repressor is going to bind. If the body's not making enough mRNA for whatever cycle or chemical reaction we're talking about, then the repressor is going to bind. So just like we saw the trip operon being negatively regulated by tryptophan molecules, there are other proteins that bind to the promoter sequences that act as positive regulators to turn genes on and activate them. So for example, when glucose is low, in E. coli especially, what the E. coli are going to do is they're going to try to find some other sugar resource for fuel. And in order to do this, the new genes to process the alternate sugars must be transcribed. So when glucose levels drop, cyclic AMP, or CAMP, is going to start to accumulate into the cell. And the CAMP molecule is actually a signaling molecule that's involved in glucose and energy metabolism in E. coli. And when CAMP accumulates, it binds to the positive regulator, or the CAP, the catabolite activator protein. And this particular protein has the capability to bind to the promoters of operons, which control the processing of alternative sugars. So they help E. coli find an alternative energy source. And then when CAMP binds CAP, the complex then binds to the promoter region of the genes that are needed to use the alternate sugar sources. And that's what you see in the picture here. So I'm going to go over the picture again too after this explanation. And in these operons, a CAP binding site is located upstream of the RNA polymerase binding site in the promoter. This CAP binding actually stabilizes the binding of RNA polymerase to the promoter region, and it actually increases the transcription of the associated protein coding genes. So what we actually see in this picture is we see the transcriptional activation by the CAP protein. So again, when glucose levels fall, E. coli needs to find some other energy source. And in order to do that, E. coli is going to start to transcribe new genes. So as the glucose supplies become limited, the CAMP levels are going to increase. That's really important. Glucose is limited, CAMP levels start to increase. And then CAMP is going to bind to the CAP protein. So you see the binding here, CAMP binding to CAP. And CAP is a positive regulator that binds to a promoter region upstream of the genes required to use the other sugar sources. So it's going to bind to this promoter, RNA polymerase is going to come in, and then we're going to allow for transcription to occur. So in this particular example, we are synthesizing and producing lactose transcripts. LAC-A, LAC-Z, LAC-Y, lactose. And the next type of operon we're going to talk about is the example of the inducible operon. So remember what I said about the inducible operon. It can either activate or repress, depending on what the energetic demands of the cell are. So we're going to talk about the LAC operon, or the lactose operon. So this is an example of an inducer. And in the absence of lactose, the LAC repressor binds the operator and transcription is blocked. In the presence of lactose, the LAC repressor is released from the operator and transcription proceeds at a very slow rate. So there's lactose signals. When lactose is signaling up here, we're going to have blocking of transcriptional processes. In the presence of lactose, well, then the repressor has been bound with lactose and it's going to be released from the operator. So here you see the binding of the operator. When the repressor is bound to the operator, nothing can happen. But in this picture down here, there is no relationship between the repressor and the operator. So the operator, it can operate, it can produce the transcript. So it's producing that lactose transcript in this picture. But the LAC operon is actually pretty complicated. And in order for it to be properly activated, there must be two conditions that have to be met. So first, the level of glucose must be very low or non-existent. And second, lactose must be present. We just talked about that on the previous slide. And only when glucose is absent and lactose is present will the LAC operon be transcribed. In the absence of glucose, what is actually going to happen is the binding of the CAP protein makes the transcription of the LAC operon more effective. So that's what we see in this picture here. So when lactose is present, its metabolite, allolactose, binds to the LAC repressor and it changes its shape so that it cannot bind to the lac operator and prevent transcription. That's what we actually see down here. So the repressor is binding. So again, anytime that we have the repressor bound to the operator, it's repressing the operator, it's repressing operations. It's not going to carry out. So these different conditions, they make sense for the cell because it would be energetically wasteful to synthesize the enzymes to process lactose if glucose was plentiful or if lactose was not available. And you also be aware that the lac operon is transcribed at a very, very low rate, even when glucose is present and lactose is absent. So really important. So again, up here, transcription is occurring. The CAMP-CAP complex is stimulating the RNA polymerase, and the RNA polymerase is allowed to act because there is nothing bound to the operator. The operator's free, and this is going to increase RNA synthesis. Down here, even in the presence of that CAMP-CAP complex that is meant to induce the mRNA transcript production, the RNA synthesis is blocked in this example because of the repressor. And again, just reinforcing some really important concepts about the complexity of eukaryotic gene regulation. It's much more complex than we see in prokaryotes because there are potentials for epigenetics to occur. Our genes can change as we grow older. Our genes can be modified by the environment. The field of epigenetics is a booming field, and there's a lot of research about the processes of DNA methylation, histone methylation, and how these little processes that are very intricately woven can change gene and protein expression. Very, very interesting. Look up epigenetic regulation and epigenetic modifications if you're interested. So again, eukaryotic organisms, this first bullet point, transcription and translation are physically separated. Different than what we see in prokaryotic organisms. In prokaryotic organisms, we see those processes occurring simultaneously. And in the eukaryotes, regulation can occur at many levels. So we can have post-transcriptional modifications, we can have post-translational modifications, and the protein that depends on all of these modifications can even be regulated itself. The degradation mechanisms of proteins can be influenced by lots of different chemical signals. So again, the first level of eukaryotic gene regulation begins with those modifications to the DNA. How is the DNA being modified? And the epigenetic regulation is going to be responsible for that. How are the histones being methylated? What's happening with methyl groups being added to and taken off histones? And again, this whole process occurs way before transcription even begins. And the transcription factors, they're very important for gene regulation as well, as are the proteins. So the transcription factors, the post-transcriptional modifiers, and the post-translational modifiers are going to result in how that final protein is actually expressed. So we've actually sequenced the entire human genome. Really interesting project. Google search the Human Genome Project. So we've discovered that a human being is made of approximately 20,000 genes. We get 23 chromosomes from our mom, 23 chromosomes from our dad, and our DNA is wound, compacted, and intricately placed in our body, and our DNA has particular modifiable factors. These are going to be the histones, like I've talked about. The histones can undergo the epigenetic regulation, the methylation in particular. And whatever genes are expressed, they must be unwound and they must be made available for polymerases. Remember, RNA polymerase is going to bind to that operon, and it's going to basically start the whole process of mRNA transcription. So this is a really cool picture of how DNA and proteins are actually organized. So here we see the histone, and we see DNA tightly wrapped around the histone. So you can imagine, as the histone is becoming modified, as we're getting all these methylation players and different chemical sequences coming in. If the histone changes, well, what's around it may be susceptible to change as well. So the DNA is actually folded around these histone proteins to create what's known as nucleosome complexes. And these nucleosomes control the access of proteins to the underlying DNA. So the DNA around the histone is the nucleosome. So all of this is a nucleosome. And this is a really cool picture of an electron microscope that we see here. We're visualizing what DNA looks like on an electron microscope, what the nucleosomes actually look like. So they actually look like beads on a string. How closely the histone proteins associate with the DNA is regulated by signals that are found both on the histone proteins and on the DNA. So remember back to that previous picture, we saw the histone and we saw how the DNA is wrapped around it. And these signals are functional groups added to histone proteins or to the DNA, and they determine whether a chromosomal region should be open or closed. And there are particular tags that happen, and these tags, they're not permanent, but they can be added or removed as needed. So these are the histone tags. Some chemical groups, such as phosphate, methyl, or acetyl groups, are going to be attached to the specific amino acids in the histone tails, right here. And these are located at the N-terminus region of the protein. And although these groups don't alter the DNA base sequence, they do alter how tightly wound the DNA is around the histone proteins. So remember, DNA is a negatively charged molecule, and unmodified histones are positively charged. So we have a negative and a positive working together. So the changes in the charge of the histone will change how tightly the DNA molecule will be. By adding chemical modifications like acetyl groups, the charge becomes less positive, and the binding of DNA to the histones is relaxed. Altering the location of nucleosomes and the tightness of histone binding actually opens some regions of chromatin to transcription and has the capability to close others. So again, I mentioned on the previous slide, DNA can be modified by methylation, the addition of methyl groups. And this methylation process occurs in very specific regions called CPG islands, and we're going to talk about those on the upcoming slides. So I mentioned the CPG islands on the previous slide. I just kind of want to finish up saying what I had to say about them. They are stretches with a high frequency of cytosine and guanine, CPG, dinucleotide DNA pairs. So they have high concentrations of cytosine and guanine, and they're found in the promoter regions of genes. So the cytosine member of the CG pair can be methylated. So that's just when a methyl group is added, when that CH3 group is added. Methylated genes are usually silenced, but sometimes methylation can have regulatory effects. In some cases, genes that are silenced during the development of gametes of one parent, they can be transmitted in their silent condition to the offspring. And such genes are said to be imprinted. Parental diet or other environmental conditions may also affect the methylation patterns of genes. And there are actually research studies today that show our diet and environmental factors can alter our genes, hence the word epigenetic modifications, epi meaning above the genes. So changes in chromatin organization interact with DNA methylation. DNA methyltransferases appear to be attracted to chromatin regions with specific histone modifications. And there are some regions that are highly methylated, also known as hypermethylated DNA regions. And these regions contain deacetylated histones, which are tightly coiled and transcriptionally inactive. So in this picture here, we look at some of the potential consequences of epigenetics. So epigenetic modifications to the extreme can actually cause cancer, autoimmune diseases, and mental disorders, as well as diabetes. There are a lot of studies that show epigenetic modifications, especially associated with our diet, can cause all of those conditions. And so the unwinding and the opening of DNA allows for the transcription factors to bind to the promoters and other regions upstream, and that actually causes the transcriptional processes to be initiate it. So again, this picture is just showing how histone proteins and DNA nucleotides can be chemically modified, and the modifications affect nucleosome spacing as well as gene expression. As we mentioned previously, eukaryotic gene transcription and regulation is more complex than prokaryotic gene transcription and regulation. And eukaryotes, in order for the genetic transcription processes to begin, RNA polymerases are needed. They have to bind to that operon region. And they require specific transcription factors, these specialized proteins, to initiate the transcription sequence. And these factors, they bind to that promoter sequence like I talked about, and they can also bind to other DNA regulatory sequences. And the RNA polymerases require transcription factors in order to initiate transcription. This is a really cool video of an example of transcription regulation. It talks about the insulin signaling and how insulin will cause different reactions to happen in the body. So please take a minute to pause the PowerPoint and check this video out. Now that you know genes are organized in a particular fashion in order to control how gene expression works, let's talk more about the transcriptional machinery. So let's begin by going over a few definitions. The first one we're going to talk about is the promoter. So the promoter region is located directly upstream of the coding sequence. And it can be relatively short, only a few nucleotides in length, or it can be really, really long, several hundred nucleotides in length. And just a general rule of thumb, the longer the promoter is, the more available space there is for proteins to bind. And this also adds more control to the transcription process as a whole. And the length of the promoter is gene specific, and it can vary between genes. And the level of control of gene expression can also vary between genes. So the whole purpose of the promoter is to bind transcription factors that control the initiation of transcription. And within this core promoter region, about 25 to 35 bases upstream of the transcriptional start site, we have what is known as the Tata box. And this actually has a consensus sequence of going from the 5' to the 3' direction, TA, TA, and then 2 additional adenine groups. So we just call it the Tata box. The Tata box is the binding site for a protein complex called TF2D, or transcription factor 2D. And this contains the Tata binding protein. And the binding of transcription factor 2D recruits other transcription factors, including transcription factor 2B, 2E, 2F, and 2H. And some of these transcription factors actually help to bind the RNA polymerase to the promoter, and others help to activate the transcription initiation complex. You don't need to be able to differentiate between the different transcription factor 2s, so you don't need to know the difference between 2B, 2E, and 2F. I just want you to know their general purpose. So again, they are transcription factors to help bind the RNA polymerase to the promoter, and they can help activate the transcription initiation complex. This picture actually shows the interaction between proteins at the promoter and the enhancer sites. So we haven't talked about the enhancer sites, so let's go over that really quick. An enhancer is a DNA sequence that promotes transcription, and each enhancer is made up of short DNA sequences called distal control elements. So these are all the distal control elements right here. And the activators that are bound to the distal control elements can interact with mediator proteins and transcription factors. So these activators here, they have the potential to activate the distal control elements and enhance the enhancer's capability of starting the entire process. Two different genes may have the same promoter, but different distal control elements, which may enable differential gene expression. So again, the transcription factor 2D, that's going to bind the Tata box. And when transcription factor 2D binds that Tata box, what that's actually going to do is that's going to recruit additional transcription factors to form a complex at the Tata box. So then what could happen is RNA polymerase is going to bind upstream, and then it actually gets phosphorylated by different types of kinases, and part of the protein is released from DNA. RNA polymerase is in proper orientation for transcription, and then we can transcribe whatever mRNA sequence that we want at that point. So again, just reiterating some important complexes, going back over the enhancer. So I know this is redundant, but when I make slides redundant, it's because these are really important concepts and you may see them on quizzes and exams. So again, the enhancers, they enhance the transcription process. These distal control elements bind to these activators, which again facilitate the enhancement of the initial starting sequence. And remember, two different genes may have the same promoter, but different distal control elements, and they can enable differential gene expression. And let's talk briefly about this DNA bending protein. This is responsible for changing the shape of the DNA and properly orients the enhancer region. So in order for the enhancer region to be properly configured and for this whole process to happen, the DNA actually has to be bent in a perfect conformational shape in order for this to occur. If that doesn't happen, RNA polymerase is never going to bind to the promoter region, and then transcription is never going to proceed. And a very important concept that we've talked about briefly is reiterated on this slide. So before we leave the process of mRNA transcription and start translation, all of the transcript processing has to cease. It has to be completed, and that process has to be terminated. There cannot be any processing of RNA transcripts prior to the post-transcriptional modifications. There cannot be any RNA transcripts that are being processed prior to translation. Again, this is a huge concept that's different from how prokaryotes undergo transcriptional gene regulation. Prokaryotes can do simultaneous transcription and translation. Eukaryotes, it's very segmented and in a proper sequence because the process is much more complex. And because the process is much more complex, we're able to regulate our gene expression in a much more orderly fashion compared to prokaryotic organisms. In eukaryotic cells, the RNA transcript often contains regions known as introns and exons, and we've talked about these. So in the pre-mRNA molecule, you can see 1, 2, 3 introns, and we can see 1, 2, 3, and 4 exons. So again, the introns are going to be removed prior to translation. They're going to be spliced out by those spliceosomes. And the regions of RNA that are actually going to code for the proteins that are going to be involved in the expression sequence, those are the exons. So again, after an RNA molecule has been transcribed, but prior to its departure from the nucleus to be translated, the RNA is processed and the introns are removed by the spliceosomes, by the process of splicing. And remember, spliceosomes are ribonuclear protein complexes that can recognize the two ends of the intron, cut the transcript at those 2 points, and bring the exons together for ligation. And that's exactly what we see here. So there's still some scientific debate on how splicing actually has evolved over the years, how it originated, but there are some things that many scientists are in agreement with. So we know that splicing requires the proper identification of introns. We know that spliceosomes do the splicing. We know that if there's any errors in this process, that we could potentially splice out the wrong thing. We could splice out an exon, which would result in an altered gene expression, altered protein sequence. That may be very bad to the organism, and it could produce some severe mutation. But it could also be a good thing. If we lose an exon that's coding for some deleterious disease or some debilitating pathology, well, that new variant might actually have an adaptive advantage. So again, we still don't understand the whole process of splicing and how the spliceosomes just know what to do. And we don't understand the evolutionary processes of how spliceosomes have evolved to, in some people, to help delete all of those bad pathologies and why they include some of the non-coding sequences in some individuals. It's still something that's left up for a lot of debate, and a lot more understanding and research has to be done. So RNA, working with RNA is a very challenging process. RNA is not a very stable molecule. And It's not a very stable molecule, so if you leave it out at room temperature for over an hour, it's going to be completely degraded. And the reason it's not stable is because it undergoes this precise regulatory mechanism of control. There's a lot of players and a lot of things can go wrong. So before mRNA leaves the nucleus, it's given these two protective caps. So it's given a 5' cap of methylated guanosine triphosphate, or GTP, and that is thought to help prevent against the degradation of the transcript. So we see this cap here, right upstream from the 5' untranslated region. And then we also get a cap just a little bit downstream from the 3' untranslated region known as the poly A tail. And this is just a repeat of adenines. So you see just A. So the poly A tail also plays a role in helping to prevent the degradation of the transcript. And the protein coding region, so all the coding is contained within the exons. We've talked about that already. It's flanked by a 5' untranslated region and a 3' untranslated region. And we see all the RNA binding proteins bind to the 3' untranslated region. So they can bind to the 5' as well. So you do see that here. But the RNA binding proteins at these untranslated regions, they are predominantly, in addition to the caps, they are predominantly what's responsible for the RNA stability. So they can either increase or decrease the length, the time mRNA is present in the cytoplasm, and they also regulate the mRNA localization and protein translation. So again, if they're not present there, it can be transcribed too quickly, transcribed too slowly, and that can result in altered protein expression. MicroRNAs are very interesting, and actually my first scientific paper that was ever published was one that investigated the microRNA's influence on the stability of the circadian clock. And we found that if we deleted microRNAs, well, then we could actually accelerate the circadian clock, which eventually was a result of altered protein expression. So in addition to RNA stability, there's a lot of other different classifications of RNA. So we talked about the tRNA, the mRNA, I mentioned briefly the siRNA, small interfering RNA, but now we're going to talk about microRNAs. So again, just like all the other RNAs, these can bind to RNA molecules, and they are short. They're only about 21 to 24 nucleotides in length, and there are pre-microRNAs that are actually chopped into mature microRNAs by a protein known as dicer, D-I-C-E-R. And like transcription factors and RBPs, mature microRNAs recognize a specific sequence and they bind to RNA. But microRNAs also associate with a ribonucleoprotein complex called the RNA-induced silencing complex, or RISC. And the RNA component of the RISC base pairs with complementary sequences on an mRNA, and they either impede translation of the message or they lead to degradation of the mRNA. So what is this saying? I know that's a very complex explanation of all of that. So basically all I want you to know is that microRNAs can either accelerate transcription or they can delay transcription. Just like we saw in transcription, translation is also controlled by proteins that bind and initiate the process. So in translation, what's actually happened is the complex that assembles to start the process is referred to as the translation initiation complex. In eukaryotes, translation is initiated by the binding of the methionine initiator tRNA, this complex right here, to the 40S ribosome. This tRNA is brought to the 40S ribosome by the protein initiation factor eukaryotic initiation factor 2, or EIF2. So again, EIF2 is the first protein to bind and form the complex in translation. Really important bullet point. Definitely ask a test question on that. So then GTP plays a role. So the EIF2 protein is going to bind to GTP, and then that complex binds to the 40S ribosomal unit. And once that complex is bound to the 40S ribosomal unit, a second complex forms on the mRNA. And several different initiation factors are going to recognize that 5' cap of the mRNA molecule, and proteins bound to the complex together with the 40S ribosomal complex. The ribosome then scans along the mRNA until it finds a start codon. And remember the start codon? Methionine. What's the sequence for methionine? I hope you're saying AUG. So that's methionine. Don't get methionine confused with the stop codons. UAA, UAG, UGA. So when that mRNA finds that start codon, that methionine group, we can start this process. When the anticodon of the initiator tRNA and the start codon are aligned, GTP can then be hydrolyzed. GTP can then be hydrolyzed. And then the initiation factors are released. So GTP is converted to GDP via the process of hydrolysis. So after that happens, the large 60S subunit of the ribosome, the 60S ribosomal subunit, can bind to form the translation complex. The binding of the EIF2 to the RNA is controlled by phosphorylation. If EIF2 is phosphorylated, it undergoes a conformational change. Really important to know, I want you to know what molecules are responsible for phosphorylation. Phosphorylation is mediated by specific molecules known as kinases. Kinases donate a phosphate group and they cause phosphorylation to occur. Kinases. So again, if the EIF2 molecule is phosphorylated, it's going to undergo that conformational change and it cannot bind to GTP. Therefore, the initiation complex cannot form properly and translation will be impeded. When EIF2 remains unphosphorylated, the initiation complex can form normally and translation can proceed. So the kinases act somewhat like the suppressor molecules or the repressors do. So this is just a schematic representation of what I talked about on the previous slide, but really important concept, so please make sure that you understand this. When the EIF2 molecule is phosphorylated, a conformational change occurs. That conformational change is counterintuitive. It's counterproductive to translation. So again, when it's phosphorylated, that's the donation of a phosphate group by a kinase. It's going to block translation. Translation does not occur. No translation. When the EIF2 remains unphosphorylated, so we don't see the phosphate group down here, translation will occur. So phosphorylation impedes translation. Phosphorylation of EIF2 impedes translation. And I mentioned briefly on previous slides that proteins can be chemically modified. So we talked about the ubiquitination, sumylation, methylation. These are all post-translational modifications that can happen. And these chemical changes, they regulate protein activity or the length of time that proteins can exist. So these changes can alter epigenetic accessibility. They can actually have an effect upstream on transcription. They can change mRNA expression, transcription as a whole, or they can dysregulate translation. And again, they're all going to cause potential alterations in the expression of genes. So again, like in the circadian clock, I mentioned it was a negative feedback loop. So if one loop has post-translational modifications, that's going to have an effect on gene expression of the next cycle. So something that has a post-translational modification can affect the gene and mRNA transcription on the next cycle. So I studied in-depthly ubiquitination. So these ubiquitin molecules build up on a protein over the course of our day. So specifically in the circadian clock, there's a specific ubiquitin that builds up on the period proteins. And as this chain builds up more and more and more, well then what that does is that allows for the proteasome, one of the degradation machineries of our body, to recognize this protein that is highly ubiquitinated, and then it breaks down that protein to form amino acids eventually. So again, when we tag it with when the protein's tagged and has a multi-ubiquitinated chain, it marks it for degradation. The proteasome is going to break it down, and then once our protein's broken down, the circadian clock can start over. New mRNA transcripts can be formed, and then new protein can eventually be translated. Now let's switch gears from translation and let's talk about when cells don't undergo transcription and translation as they should, like we see in cancer. So really important thing I want you all to know is that cells die one of two ways. They either die via apoptosis or necrosis. Apoptosis is programmed cell death, and necrosis is a result of ischemia or lack of oxygen to a tissue over an extensive period of time. If our tissues don't get oxygen, they become hypoxic or oxygen deprived, and then they eventually become anoxic. They don't have any oxygen supply at all, and they die off. So with apoptosis, that means, again, programmed cell death. So with programmed cell death, our cells are designed to, well, our mitotic cells, our somatic cells, are designed to be turned over. They only last for a certain period of time. For example, our red blood cells. So our erythrocytes, they are supposed to be turned over every 120 days. And there are some diseases where the erythrocytes are turned over too quickly. In some cases of sickle cell anemia, they're turned over as quickly as 20 to 40 days. And the anemia is induced by the loss of the red blood cells. The loss of the red blood cells, you all know that hemoglobin is attached to those red blood cells. Hemoglobin transports oxygen, and the tissues in sickle cell anemia patients don't get the oxygen that they need. So they can go into a lot of sickle cell crises. But cancer as a whole, it's not a single disease, but it's an accumulation of many different diseases, actually. So cancer occurs when cells do not undergo apoptosis. They don't die when they should, and they become anti-apoptotic. So cancer cells actually kill our good tissue via necrosis. They are anti-apoptotic, and then they go into our tissue. And cancer as a whole, tumors, they can develop their own blood supply. So they can actually take blood, they can take oxygen from the tissue. And our good healthy tissue can be killed off by cancer cells. And cancer cells are thought to be caused by mutations. Mutations that modify the cell cycle control, that don't tell the cells to die off as they should, and they just continue to grow and to grow and to grow. There are several really important tumor suppressor genes that we have in our body. And as the name implies, tumor suppressor genes, they suppress inappropriate cell growth. And there is one in particular, tumor suppressor gene P53. That just means protein 53. If you run an immuno-western blot, this protein can be isolated at 53 kilodaltons. And this is a tumor suppressor gene that's mutated in over 50% of cancer types. So again, if this undergoes a mutation, it doesn't have its tumor suppressing properties and tumors can form. It actually functions as a transcription factor. And then we have oncogenes and proto-oncogenes. So proto-oncogenes are positive cell cycle regulators. They protect us against cancer. Oncogenes are the genes that cause cancer. Oncology is the study of cancer. So oncogenes are genes that cause cancer. So this histology slide actually shows a particular type of cancer known as Burkitt lymphoma. And what's happening here is the B cell lymphocytes undergo lots of mutations and cancer arises. So again, cancer is caused by mutated genes. Our proto-oncogenes may not be working as they should, and our oncogenes may be superseding the roles and the effects of the proto-oncogenes. And in particular, one of the genes that is mutated in Burkitt's lymphoma is one known as c-Myc. I'm going to add a C in here. So c-Myc. So this is a transcription factor that's activated in Burkitt's lymphoma that's not normally activated in normal tissue and normal cells. And there are a lot of mutations that can actually activate transcription factors. And on this slide, I'm just going to talk about a few of them. So overexpression of the epidermal growth factor receptor. So if we're having our top layer of our dermis grow out of control, well, that just makes sense that it could cause several different types of breast cancers to occur. This leads to activation of protein kinases and overexpression of transcription factors that control genes involved in the cell cycle. The cell cycle gets out of whack. Cells that should be dying at a certain time no longer die at that certain time, and they proliferate out of control. So if patients have an overexpression of the epidermal growth factor receptor, then there are different pharmaceutical approaches that the physicians will utilize in order to prevent activation of EGFR. And EGFR inhibitors are very successful in treating certain types of breast cancers.